Joe, want to go ahead and get started? It's 11:30 or 8:30. Or any number of times, I guess, for all of us.
Yes, anytime. Sure. Jose can start.
Okay.
Slowly, maybe.
All right, welcome to WNAR 2022, and this is the session Adaptive Design and Clinical Trials. Dr. Moeller is our first presenter, so we'll let him take it away.
Okay. I'm going to share the screen. Okay, it's okay. Do you see it okay?
Yes.
Yes.
Yeah.
Well, so thank you very much, Jessica, and all of you. The title of this talk is Designs for the Joint Estimation of a Target Dose and its Slope at the Dose Response Function. This is a joint work with Nancy Flournoy from the University of Missouri, Hyun from the Tempus Lab, and Fernando Plo from the University of Zaragoza, and myself, Jose Moller, from the Public University of Navarra in Spain. Well, first of all, I'm going to give you the framework of this talk that will be clinical trials. Here in this graph, we see the doses, the dose levels in the x-axis, and in the y-axis, the probability. And here is the toxicity function, which is of interest in this talk. Our main goal is to estimate the dose with a toxicity target, for instance, 0.3 in this case. And we are going to assume that the toxicity function increases with the dose, okay? So we are very interested in estimate this dose, and any bias is risky because we can apply more toxicity than allowed, or if we underestimate the dose, we can allocate patients in doses with a low efficacy, okay? So why the interest in estimating in the joint estimation of the dose and the slope? With this graph here, we have a set of logistic functions. All of them have the same target dose, is near the 0.7, and all of them have a toxicity 0.3. But the difference is the slope. As you can see, the slope, do you see my pointer? My pointer, yeah? Okay, the slope in a logistic is proportional to the beta parameter, okay? And here, gamma, capital gamma, is the toxicity target. Okay, so when we have very flat slopes, any bias in the estimation is doesn't have important consequences, because if we overestimate, maybe we apply more toxicity, but we don't pass very much our target. But if we have very large slopes, is the green case, any overestimation of the target dose will be very risky for the patient, because he can have a level of toxicity really dangerous. So the slope can be seen as a risk, or a measure of the risk, of an ethical measure about the importance of the right estimation of the target dose. From the point of view of optimal designs, in this work that is here by Ford, Thorsney, and Hu, everything was said. In the case of the logistic, we know that the optimal design to jointly estimate the target dose and the slope, we know that the optimal gives the minimal global variance, we can say in this way, to estimate the dose, the target dose, and the slope. So in this case, we have to allocate half of the patients in this percentile, I mean, in the dose with a probability of toxicity 0.1, and the other half of patients in the dose with toxicity 8.2. So this is solved, but optimal designs have very important problems to be applied in practice. First of all, to have the optimal design, we need to know very well the response function. We need to know if it is logistic or probit or exponential or whatever. Even if we accept that the response function is logistic, we need to estimate the parameters because the optimal design in this case depends on these parameters. Third, even if we know the parameters, the optimal design, the main target is to estimate the global response function, but our interest is in a specific dose. And finally, another practical problem, very important, is that the optimal designs tend to allocate patients in quite extreme doses. For instance, this dose is not possible in practice because the level of toxicity is really high. So there is no global solution to these problems, but there are partial solutions. First, response adaptive designs. Response adaptive designs allocate patients in doses that try to be close to the target dose. There are two different kinds of response adaptive designs in this context. First, the up and down designs, and a very important one is the 2RD, and Bayesian designs that have a prior function of the toxicity, and then with the a posteriori distribution, they obtain the dose with the toxicity target, and the following patient, they are sequential designs, so they apply one dose to one patient, and when we have the response, they allocate the following patient. So in each step, they allocate patients in a dose which is the closest as possible to the target. Okay, but this kind of designs that try to concentrate allocations close to the target dose don't converge with probability 1 to the target, and this is proved in this paper by Adriel. Okay, so the main advice they give is that this is a good idea to allocate patients knowing one dose, but in the two closest doses to the ones to the estimated target dose. Also, the same advice is given in a seminal paper by Wetherill that it's better to allocate patients not in one dose, but try to allocate patients in doses that are close to the target. Okay, the estimated target, of course. Another partial solution are restricted designs that is given in Biederman et al. And Haynes and Rosenberg. The idea is to avoid the allocation of patients over a level of toxicity that we fix. But this level of toxicity should be higher than the target because we need to have information around the target dose. Finally, in order to avoid problems of misspecification, another advice is to use non-parametric estimations, for instance, the isotonic estimator in our case. Here, I present to you our proposal. One proposal we have to estimate jointly the target dose and the slope is the neighborhood designs. Neighborhood designs, I'm going to focus on the simplest expression, which is this one. They are a general model, but I'm going to focus on that in this talk. The idea is to allocate patient. Mu will be the notation for the target dose, and delta will be called the radius, and it's very you will see later, but it's related with the slope. Okay, so we allocate half of the patients on the left of the target dose and the other half on the right of the target dose. Okay, so with this expression, this theoretical expression of a neighborhood design, first we're going to study the theoretical properties. In order to do that, we are going to consider the link function, this link function. I mean that we are going to change from a space dose to this and to this other space. We are going to move the dose space. And for this list of response functions, that is not exhaustive, we can give a larger list of response functions. We are going to reparameterize the distribution function. For instance, in the logistic, these are two classical expressions of the logistic function. We are going to reparameterize this, and we are going to consider another distribution function, this equivalent, but in the new space of doses. Okay, but in this case, the mu is the target dose, the dose with toxicity capital gamma, and eta is the slope in the target dose with this response function. Okay, so in the new space, the neighborhood designs becomes this one. And here we see that in both streams, in both points, in both design points appear the slope. And also we see that the optimal properties of a neighborhood design depend only on this parameter A, which is the product of the slope and the radius. I'm not going to explain this, but only I'm going to say you that following Biederman and et al. Results, we are able to write in a very easy way the determinant of the information matrix associated to the neighborhood design. Of course, to obtain, to be valid this expression, we need to use this reparametrization. Okay, and here we see that the optimal number of patients in each design point is 1/2, and also we see that the maximum value of the determinant of the information matrix is reached in an optimal value A star that can be calculated exactly for each of the response functions that we have in this list that, as I said before, can be larger, okay, but it's a mathematical exercise, not more. So we know very well some theoretical expressions, the theoretical properties of the neighborhood design. And also I wanted to show you in a simulation, a small simulation study, that neighborhood designs, at least the theoretical neighborhood designs, are competitive with the classical designs. Here we see in the x-axis several slopes, very flat slopes, very steep slopes, intermediate slopes, and here we have simulated 1,000 times 1,000 clinical trials, each with 100 patients, and at the end we have obtained the maximum likelihood estimator of the slope. As you can see, the blue one is the theoretical neighborhood design, and the performance is very good. It's almost competitive with the optimal design, and also competitive with the two classical response adaptive designs, which are the 2RD and the CR. Okay, so it makes sense to study this kind of designs. Finally, to resume, to summarize the properties, I wanted to say you that whatever, regardless the response function that we use, we have that the optimal neighborhood design, the optimal neighborhood design depends on an expression which is the product of the radius and the slope. So when we are working with very large slopes, a small radius is needed. When we are working with a very flat slope, we need a large radius. But we know that this hyperbolic relationship advises us to use values close to the vertex, so close to the 0.5. This is an advice, is not approved, but is I mean that if we have a very large slope, if we increase very much the radius, we don't have we don't perform better. I mean, because the increase of radius is very small to be optimal. But I wanted to say, finally, once we know very well the theoretical properties of neighborhood designs, that we can do the same criticism as we did for the optimal, because the theoretical neighborhood design depends on the parameters, on the unknown parameters. So we cannot apply directly them in practice. This is why we propose adaptive neighborhood designs. Adaptive neighborhood designs means that we have one of the following situations happen. First, maybe we know we have some previous information and we know where the target dose is, not exactly the value, otherwise it doesn't make sense to do statistics, but where is it's the value, in which interval is. Otherwise, we can propose a startup rule to do a first estimation and then to propose a neighborhood design, okay? Or, well, we can start in some guess and then we can, once we have allocated 50 patients, we re-estimate and we allocate the rest of patients with a new re-estimation. So in order to apply neighborhood designs in practice, we need to do a first estimation in order to work with. So neighborhood designs doesn't work with the real values, but with some initial estimations that we are updating in several steps, okay? Well, we have done a deep study, simulation study, and we have obtained some conclusions. First, the best performance for the neighborhood designs requires some knowledge of the target dose. And a way to solve this in practice is to use to warm up the model or to use a startup rule. We know from a previous work that the best startup rule is the 2RD and response adaptive designs, but we need to use a very small number of patients. In general, we proved in a previous work that with 10, 15 patients is enough. And also, we know that when we are using a very small number of patients, we are not able to obtain with a high probability the maximum likelihood estimator of the target dose, which is very important to have in order to start the adaptive neighborhood design. So here we see that at least we need 20 patients to have with a probability of 80% the maximum likelihood estimator of the target dose when we have very flat slopes or very large slopes. So we will use the isotonic estimator that is more reliable, at least to estimate an initial number of patients. And here I'm going to skip this and I'm going to focus on this slide. These are results of a simulation. Here we have a lot of designs. I'm going to focus in the yellow ones. Here, to summarize this this table, we have simulated the adaptive neighborhood designs with 10 patients, 15 patients, or 20 patients in the startup rule, and then we have applied the neighborhood design with the initial estimations. And here, this is a neighborhood design without restriction. I mean, the neighborhood design without any restriction. This is a neighborhood design with a restriction in the maximum toxicity level allowed. Okay, this is a neighborhood design without restriction, but with 50 patients in an initial estimation, and the rest. And then when we have 50 patients, we re-estimate the values. So this has three steps: the startup rule, the first batch of patients, and then array estimation. And here the same one with a restriction in the maximum toxicity. And the blue ones are theoretical designs, the optimal design restricted to a maximum toxicity, and the theoretical neighborhood design. As you can see, it's difficult with so many numbers, but I tell you, we see that the optimal number of patients to work with in the startup startup rule is 15, 1-5, okay? And it's curious, but the neighborhood restricted design doesn't perform very bad. Of course, these values, which are mean square errors, are larger than the optimal, of course. This is this is logical, but we have smaller values than the theoretical neighborhood design. Okay, so the performance is not so bad. Of course, the performance improves as the slope is larger. Okay, I wanted to tell you that once we have to finish, I will show you another design that we have studied, because the neighborhood design, the main idea is some symmetry in the two design points we are using. But when we put a restriction, we are breaking this rule. So we thought that maybe we can think in another kind of designs where that directly maximize, designs where we put two restrictions. I mean, designs where we put an ethical restriction. We, for instance, we don't want to put patients to a dose with toxicity larger than 0.4, for instance, and we want an inferential restriction. We want to obtain a design that at least is 80% efficient with respect to the C optimal, for instance. Then in this case, we only have to maximize this product of efficiencies. And we have obtained also another design, which is this one, which is another kind of, and we call this design CC optimal designs. As you can see, it only has 2 points as the neighborhood design, but there is no symmetry. Okay, only to show you, to finish, that this new design improves the performance of the neighborhood design when we have very flat slopes, but it's not better when we have larger slopes. Okay, so as some conclusions to finish, neighborhood designs make sense when the responses are not immediate. Okay, and in order to fulfill deadlines in our clinical trial, we have to allocate patients in a short time, for instance, all at once. In this context, neighborhood designs make sense because they have good performance in this case, and in response adaptive designs are not applicable. Another point is that they require some previous information that can be obtained with an startup rule only with 15 patients. The performance, even with restriction, is not bad. We see that the larger is the slope, the better is the design. They are robust designs, and there is a specification of the toxicity response. And we have observed that there is some overestimation of the slope. I mean that they overestimate the risk of the estimation we have done with the target dose. And I think it's my time. Thank you.
Thank you very much. We have like 2 minutes if anybody has any questions.
We covered a lot.
For sure. All right, Dr. Flournoy is our next speaker.
Okay, thank you. I will share my screen. Put it over here. Okay, do you see it?
Yes. Can you make it?
Yeah, I will. Just a second. Okay, thank you very much to Suhwon for inviting me and for the caucus for sponsoring this session. I'm going to talk about likelihood-based early stopping decisions. And this is work with my co-author, Sergey Karima, who is at the Medical College of Wisconsin. Okay, well, let's see, page down didn't work. There, okay. Today's talk is based on a paper that we just had published in Metrica, and it's called Most Powerful Test Sequences with Early Stopping Options. The most powerful test sequence part is not really the most important part of the paper. The development of this likelihood framework is, and so, you know, we consider the most powerful test part an application of the likelihood part. And so I'm going to focus on the likelihood framework in this talk. We also had a discussion of a recent paper by Nigel Stollard on enrichment designs, and we used the opportunity to discuss his paper to show how the likelihood framework would apply as an alternative to combining p-values when you have different stages. And that's online now with biometrics. So to start out with, let me just point out that I'm thinking now of sequential of designs with, well, and keep in the back of your mind something like group sequential designs. You have individuals or groups coming in sequentially. And most of the theoretical work for these kinds of designs has focused on controlling type 1 error, and particular results have been developed under the null hypothesis. So, I mean, I sit on a data safety monitoring board, and sometimes you actually stop early and you want to estimate your effect size after you've stopped, not just under the null hypothesis. So you may want to do estimation period and other things with your data besides just do a hypothesis test. So that's part of the motivation. Most of the theoretical works go one step ahead, conditioning on the study continuing. And so they're not looking at the whole probability space as a unit. And most of them rely on asymptotic results. And the asymptotic results all have assumptions attached to them. And in order to get our paper published, I read, my background has been primarily adaptive designs that deal with dose changing treatment allocations. And so it's only been with the work of my collaborator that I have looked at early stopping decisions. And so I really had to go back and read a lot of literature, and I read it with the idea of there's so much of this literature concludes that everything's nice and multivariate normal. And so I go back and I read and I read and I read and focus on the assumptions. And I'll say more about that later.
Nancy, we still see your first slide.
I know. So that's happened before to somebody. I'm going to stop my share and come back then.
Okay.
Okay. Share stop.
Sorry to interrupt you.
No, thank you. It's not good. Okay, we're going to try this. Okay.
Oh, there we go. Now we're on outline. Goals for this talk.
Okay, so this was my leading talk mentioning these two papers and these
Thank you.
theoretical results. Now, the goals. We want to provide, we provide a unified likelihood framework for estimation and testing under both null and alternative hypotheses, and with and without having actually stopped early. And we are able to show that even with finite sample sizes, one can do a whole lot more for estimation and hypothesis testing than just controlling type 1 error. And so that's very useful. And that a lot can be done without doing asymptotics at all. And when you do need asymptotics, you can use, with a few number of stages, you can use classical central limit theorems rather than imposing additional assumptions. So first, I'm going to start by recalling the probability structure for two independent normal random variables. So, you can yawn there, but it's instructive. And then we introduce just a simple rule that permits the second random variable to be observed only if the first one is less than 2.18. Then we talk about the probability structure associated with with the first random variable and the second random variable if it's observed, and point out contradictions that occur if one was to claim that those are jointly asymptotic, even jointly normal, even asymptotically. So non-sequential experiments. We've got X1 and X2. They're both normally distributed with some mean and unit variance. So if only X1 is observed, one can write down the likelihood function, and it's maximized at the observed random variable. And if you had a big sample, that would be X bar. But for a sample size of 1, it's just the observed value. If you observe 2 normal random variables independently, you have a bivariate random variable defined on a probability space that the sample space is R squared. You have a Borel signal algebra, which is what allows you to do unions and intersections. And then you have your probability measure, which in this case is the bivariate normal with 0 correlation. Again, you can write down the likelihood function, and the MLE is X bar. So now, suppose only X2 is observed only if X1 is less than 2.18. Now, X2 is not just missing, it's completely impossible. So you no longer have a random, you don't have a random variable described on R2. And so one thing that we do is define the complete probability space. And in order to do that, you actually have to define observable random variables. The way most of the theories worked is they start out with these unobservable random variables and do a lot of theory, and then kind of patch it up at the end to make it work right. And so what we do is start with observable random variables. And so we recognize that the sample space is this combination of the fact that you stopped at stage 1, unioned with you continued at stage 1, and then observed your second random variable. And just that much reveals to you that you don't have a bivariate normal because it's not on R2. So also, how would you get a bivariate normal? Well, you'd have to let this critical value go to infinity, which is the basic assumption on the large deviation theory, is they push everything out to infinity, and that totally obliterates the stopping rule from the model. So this is a schematic of the support. And so this is for 3 stages, just to make it a little more interesting. But the one on the left, we have our 2 random variables, standardized normal random variables, 1 for each stage. And on the other graph here on the right, we have cumulative statistics. So the denominator is the same for both, but on the right, not the denominator, the horizontal axis is the same for both, but the vertical axis, the random variable is the sum of the 2, is the average of the 2 instead of the individual 1. And so you might think of doing your statistics conditional on having, well, you could have stopped at stage 1, in which case you've really just got this little piece of R1. You could stop at stage 2, in which case your sample space is this region. and you continue to stage 3, it's down here. So each of these are disjoint sample spaces. Now, your 2 random variables are the 2 x's and your decision to stop, which for x equals 2 is the same as the sample size. So you have everybody, so you did your first stage, you got your first random variable, and you only get the second one if the indicator function x1 is less than 2.18. So you have your random variables for your likelihood include this information about stopping. And if you do that, then you can write the total likelihood as this product. So we have 2 indicator functions, d equals 1 and d equals 2. In practice, you only end up with one of these pieces, which is what people kind of jump to immediately and therefore miss some of the of what's going on when you start taking expectations and stuff, because this d is a random variable. So if you stop at the first stage, then everybody just gets then you see the x1. If you start at the second stage, you see x1 and x2. And you can write that in sort of canonical exponential form. And in fact, this was recognized early on by some pretty prominent people to be a member of the curved exponential family. And so our thesis is that rather than try to shoehorn things into multivariate normal space, that one should take advantage of this representation and use it for many things. So in this space, then, one can write the log likelihood. And as is well known, the likelihood itself doesn't change from what is observed if you take these pieces and write them down independently. So you get x1 if you just had stage 1, and you get the average if you just have stage 2. So you say, why all the bother? Well, the fact that the support and the density change mean all the moments associated with the maximum likelihood estimates change, and the moments of other statistics change as well. The other thing is that the stopping stage is not kind of a random stopping thing. I mean, it's random, but it's informative. So actually, you have to be careful about whether you want to condition on it or not, because you're going to lose information if you do condition. You don't have to look too much at this slide. There's a lot of letters on it. What I want to point out is that Armitage, McPherson, and Rowe in '69 introduced a recursive formula to estimate what is a piece of the density of, so what I have on the left here, so say T is some statistic that, and with the little d, it's stage specific. With the big D, it's in parentheses, it's a random variable. And so this is associated with your having gotten to some stage, which is a random event. And so that density can be written as a sum of the individual stage-specific statistics. And this piece in the middle was recognized a long time ago and is used in all the popular software for finding, for calculating type 1 error and power. So again, you know, this piece of the puzzle has been around for a long time, and our then contribution is to point out that one can use it for, that one can embed it in the proper probability space. One can use it for calculating a lot of things besides type 1 error and power. So here's a look, just in terms of some pictures after some math. So this is, again, we've got just 2 random variables. And when we say unconditional, this is what you might have this is based on the density that you would have before you start a study. And so one question is whether this, rather than focus everything on what happens under the null hypothesis, there may be times you want to focus your design and your considerations of what's going to happen on everything being unknown, including the possibility that you might stop early. So on the left, we have the histogram of theta hat under the null hypothesis. And you can see that there's a large piece that looks pretty normal. And then there's this little bump, which has to do with the density if you had rejected the null hypothesis at the first stage. So what you see is that this little bump, if you're not under the null hypothesis, if theta was actually at 2.18, which was your point to reject, to actually decide whether to continue or not, this this little mass on the right is much more predominant, and that gets larger as the true theta actually gets out. So you have, at your a priori, when you're setting out to design your study, you have a very bimodal distribution, and one might think a little more about ways to use that knowledge in the design. Now, what happens if you stop after you've observed your first random variable? Well, certainly, when you make the decision to stop, you have a nice uniform random variable, and you decide if it's too large, you're going to stop. Now, having stopped, the question is, excuse me, having stopped, the question is, you want to now, you stopped, and you want to estimate the effect size. And so I think we should think about, I can't say I have come to any conclusion myself, but we should think about whether one should be characterizing that event, as in finding confidence intervals, et cetera, based on the normal distribution you just started out with, or on this distribution that's associated with having stopped. So again, we have two pictures here. So in both cases, you stopped. On the left, the null hypothesis, the theta was the null hypothesis, theta equal to 0. And on the right, we have theta equal to 2.18. So this is a truncated normal distribution, conditional on having stopped. And one can calculate the expectation and other moments from the theory of truncated normal distributions. And we see that the expected mean is definitely not 0, even under the null hypothesis. And I think, this may be, so people stopping and then reporting statistics of the data having stopped seems to me to be a bit like the problem of only publishing when you have p less than 0.05, that you'll end up with a collection of statistics that misrepresent what's going on, but my mind doesn't quite get there. I think I think that was worth some discussion. Here is the distributions under the same theta equals 0 and theta equals 2.8. This is if you actually complete both stages. That is, you've got both random variables. And so particularly if you look at the left, it looks awfully much like a normal. If you look at the right, there's a little bit of skew, but people that are used to doing practical statistics probably wouldn't let that cause them too much anxiety. But as theta gets bigger, the fact that these both really are mixtures of distributions becomes more apparent. So one should at least have that in your awareness when you go to analyze your data. Now, this is a little bit about well, a little bit about conclusions, about sort of just to say, as I've been saying, we want to do things besides control type 1 error and estimate power. And so, so we know that these random variables are distributed according to a curved member of the exponential family. That means I didn't have to pick normal. I could have done binomial or Poisson or exponential or whatever, and written down the likelihood associated with that, those distributions, and gotten exact expressions for my statistics really fairly directly and simply. I didn't need to force the whole problem into normality in order to be much more specific for this wide class of distributions. And I didn't need any asymptotics. So sums of the statistics also belong to the curved exponential family. And examples that we've worked out about how this construct is useful, as I alluded, the first one is in this Metrica paper. And we showed that you get uniformly most powerful tests in a particular construct of interim analyses. And again, it's it's somewhat of a toy problem mathematically, but it's to illustrate again how to use the likelihood construct in to do more. And the second example is a bit more applied, is more applied in that we were given a problem by Nigel Stallard to look at, which involved enrichment designs, and in his development of how to study the enrichment designs, he combines p-values from stage 1 and stage 2, and we wrote down the joint likelihood of an example, one of the things that he was studying, and then showed that with the likelihood construct, we could examine a wider class of alternatives and hypotheses tests than one could do by just the combining p-values approach. So these are, as I say, examples of why taking the likelihood approach is helpful and informative. Now, I keep talking about prior work depending on asymptotic normality, and I aside from the papers that I mentioned that recognize the curved exponential family and then seem to be promptly forgotten. These are some comments about the assumptions in the work that is out there for group sequential designs. And I should say that the theory here, starting with Ciatis, is really follows directly on the, I got about a minute, huh? I'm almost finished. Follows almost directly on the theoretical development for survival analysis. And in that context, they saw the stochastic process, evolves over time, and you may only see pieces of that time trend, but you have nested sigma fields. That was a big recognition for the Martingale central limit theorem work that was applied. So it turns out, however, as I tried to show you up front, we do not have nested sigma fields for this problem. And so the assumptions that people make really lead you to the conclusions of independent increments and normality that they then seem surprised to have have found. My big conclusion is that the complexity that I've just presented is not the end of the world. That is not having normality. It would have been back there in the early days. You know, we needed normality in order to be able to do anything. But we are now in a place where my colleague, Sergey, can, you know, wing out 10 to the 6 simulations while he's eating his lunch and do what until recently has been considered quite amazing things. So lots can be simulated, lots can be done numerically. The analysis presented that we're suggesting is not really, it'll be handy to have some R code, but it's not really difficult now. So working with the observed random variables allows us to do proper approximate probability descriptions without resorting to asymptotics. We partition the density according to possible stopping stages. And we have a lot fewer assumptions than others. So that is my talk. And I think I'm finished.
Thank you.
I went too far.
All right, our next speaker is Dr. Frieri.
Yes, let me share. Do you see this? Oh no, you should. Do you see the slides?
Yes.
Okay. Let's switch to. I am trying to switch into the full screen mode. Okay, can I see me moving slides?
Yes.
Okay, great. Okay, so first thing, I want to thank Suhwon for the invitation to this session. And I'm Rosamarie Frieri. I'm a postdoc researcher at the Department of Statistics, University of Bologna. This is a joint work with Bill Rosenberger, Nancy Flournoy, and Suhwon Lee. And we started this work when I was a visiting postdoc fellow at the George Mason University this fall. This talk is about the design of a enrichment trial. So when we have a new drug that has to be tested, we usually have a clinical trial in which a large group of patients is enrolled with the objective of testing the drug on a heterogeneous group of patients, because then the intention is to treat the whole population. The underlying assumption is that the treatment effect is homogeneous across all patients. However, if this homogeneity assumption is violated, the trial may be stopped with the incorrect conclusion that the new drug is not effective. And this may be because the treatment effect is attenuated due to a small group of patients that don't benefit from the treatment. Thanks to the recent advances in disease biology and biomedicine, is reinforcing the idea that not all patients can be successfully treated with the same therapy. And this is leading to what is called personalized or precision medicine. In a nutshell, it consists in giving different treatment to different patient subgroups. When there is prior evidence that the effect of a treatment may be different in certain patient subgroup, enrichment design is an efficient approach to target the patient subpopulation. This is also supported by Food and Drug Administration guidelines, according to which enrichment strategies can increase the ability of a trial to detect an effect of the investigational drug. So, enrichment design is an efficient approach for precision medicine. In this kind of trial, usually subpopulation is selected to be enrolled in the trial based on one or more biomarkers, and then the safety or the effectiveness of the drug is evaluated in the selected population. We will call that selected population target population in what follows. Probably the idea of identifying biomarker and targeting clinical trial in subpopulation was originated in the oncology literature. Then Simon and colleagues formalized the concept from a statistical viewpoint. What we want to focus on is to a paper from FDA statistician in which they insist that even if an efficient two-stage design is used to identify the subpopulation, and then at an interim analysis, the enrollment criterion changed only to target that subpopulation, still a follow-up confirmatory trial is necessary on the subpopulation. And this provided strong support to our approach, as we will see in the next slides. So the subpopulation is defined by means of biomarker, and the accuracy in the identification of the target population depends on the prior knowledge of the candidate biomarker. Biomarker can be prognostic and predictive. A biomarker is prognostic when it's correlated with patient outcome, independently to the treatment received. A biomarker is predictive when it's correlated with both the treatment and the patient outcome. Especially predictive biomarker can be used to improve the efficiency of clinical trial. When the biomarker is binary or categorical, take for instance a genetic marker, the patient subgroups are well defined. Instead, when the biomarker is defined on a continuous scale, for instance, the cholesterol, how this patient subgroup can be identified? How to identify the target population? The answer is to define a biomarker threshold that defines the target population. Assume to be in a clinical setup in which a single continuous predictive biomarker is suspected to affect the patient response. Assume also that high responses are more desirable for patients and that larger biomarker value imply a better outcome. The scheme of our approach is summarized here, so please keep this in mind for the rest of the presentation. We have stage 1 and stage 2. In stage 1, N1 patients are enrolled and randomized to treatment or control, denoted by A or B. For each patient, we observe a continuous response, Y, and a biomarker that is denoted by X. After performing stage 1, we can use stage 1 data to estimate the biomarker threshold, and so to identify the target population. Once this is done, we may wonder if the size of the subpopulation is reasonable, and so if it's worth enriching. In the case of positive answer, we proceed to stage 2. Stage 2 involves N2 patients, but only those with biomarker value greater than the estimated threshold at the end of stage 1. In this case, patients are still randomized to A and B, so treatment and control. At the end of stage 2, we are interested in whether the target population is actually benefiting from the treatment. So at the end of stage 2, we test the treatment effect. In this setting, we provide a complete template for the design of stage 1 and stage 2. In particular, we focus in how large stage 1 has to be in order to efficiently estimate the threshold, and what are the consequences if this is not done properly. We also provide insights into how the predictive nature of the biomarker impacts the validity of the second stage and the whole study. We assume that subject observations, so YI, XI, are independent conditionally on the treatment assigned. We denote by YA the random variable describing the patient response that is assigned to A, and the same for B. Then the pair YAX is assumed to arise from a bivariate normal model with means mu y given A, mu x, variances sigma y given A, sigma x, and where rho A is the correlation between YA and X. So rho A is the correlation between patient response, and biomarker. This is a key quantity. Clearly, the same is analogous for group B. We assume equal variances for the first work, and then that X is a predictive biomarker, so that rho A is greater than rho B. We denote by theta A and theta B the vector of model parameters associated to group A and group B. For convenience, let's switch from the biomarker value X to its standardized version Z. In this case, the treatment effect in the subpopulation defined with biomarker value greater than small z is given by this expression here. What is important to note is that we have a part that only depends on model parameters and then a part that depends on the biomarker, this blue lambda z here. This is the normal hazard function, so defined as the ratio between the standard normal density and the complement of the cumulative distribution function of the standard normal. But notice that this is also the expectation of a standard normal random variable truncated at z. Now, assume that the target population is that patient subgroup in which a treatment effect at least equal to c is detected. Without going to the mathematical details, then the threshold z star is the value such that that lambda of z star is equal to this quantity here. So in practice, the problem can be restated into finding the truncation point of a standard normal random variable such that the expectation is equal to this ratio. But let's see a little bit more in detail what is C. C can be seen as a sum of two components, D and delta. D is the minimal clinical significance difference that is elicited from the investigator, while delta is the additional size effect required at stage 1 in order to obtain a conclusive result at the end of stage 2. While D comes from clinical considerations, delta arises from statistical arguments. In this graph, we can observe how the predictive nature of the biomarker affects the threshold and the treatment effect. In particular, looking at the left panel, we can see the increasing behavior of the threshold as C increases. So the speed of increasing is larger for smaller value of the difference between row A and row B. So as the color line becomes darker. If we look at the right panel, we observe the behavior of the threshold, Z star, as row A minus row B decreases. And in this case, the behavior of the threshold is decreasing in this difference, and the behavior is more pronounced for increasing value of C. So as the color line becomes darker as well. Once we perform stage 1, we can analyze data and compute maximum likelihood estimator, so theta hat A and theta hat B. Then the threshold can be estimated by simply plugging in model parameters in the formula for the threshold. So hat Z star is defined at the value such that lambda Z star hat is equal to this ratio. And now comes the question whether or not it's worth enriching. So is the size of the subpopulation reasonable? This is a crucial point in the enrichment problem, and our modeling allows us to directly assess the size of the subpopulation by means of this probability. So what is the probability of observing a patient with biomarker value greater than the estimated threshold? And due to our setup, we can compute this probability using the standard normal cumulative distribution function. So what if the subpopulation is too small or too large? The trial could be stopped after stage 1 if this probability is too small, corresponding to a too small estimated threshold. In this case, the chance of recruiting patients at stage 2 becomes very low, and the subpopulation that benefits become very small. On the other hand, the trial could be stopped after stage 1 if this probability is too large, corresponding to a too small value for the threshold. What happens in this case is that the benefiting subgroup is almost as large as the whole population, and this may also lead to the decision of not enriching. Whether or not enriching is worth the cost depends on ethical and cost considerations. For example, in the case of rare or grave disease, even if a small population benefits, it may be worth enriching and continuing the trial. Or, for example, in the case of harmful side effect, it may be worthwhile enriching to avoid exposing patients to the treatment that don't benefit. In general, it's well known that the financial cost of evaluating the biomarker needs to be taken into account in both the small and the large benefiting population scenarios. Okay, so if we decided we want to proceed to stage 2 and it's worth enriching, we perform stage 2 by recruiting only patients with biomarker value greater than the estimated threshold. Following the idea of the FDA statistician, this stage 2 is seen as a confirmatory trial designed only to target the subpopulation. In this case, the data are assumed to arise from a truncated bivariate normal model. And the interest is in testing whether the treatment is superior to the control. So we test this hypothesis and we use standard central limit theorem arguments. And in the case the null hypothesis is rejected, we conclude that the treatment effect in the subpopulation defined by the biomarker threshold HAB-Z star is greater than the minimal clinical significance difference D. So we have done stage 1 and we have done stage 2. But what about sample size? How many patients should we include in these 2 studies? We propose the following approach. So starting from initial guesses of model parameters and from an initial value for D, assuming that sigma y given A and sigma y given B are known, we consider several values for delta. Of course, for each value of delta, we have a corresponding value for C. In turn, these correspond to several candidate values for the threshold that we call Z star 0 for now. Then, given the formula for the parameters of the truncated bivariate normal distribution, and given a type 1 error rate equal to alpha, the sample size required at stage 2 to achieve a power of 1 minus beta is given by this formula. So N20 is the initial guess of N2. Okay, so for each value of delta, we have a value for the threshold and then a candidate value for N2. You can see an example here. So here we fixed alpha equal to 0.05 and the power to be 80%. And you have several experimental scenarios corresponding to different choices of the correlation rho A and rho B. And we have increasing delta here and the candidate value for the sample size of stage 2 inside the table. And it's easy to see that there is a trade-off between small delta and large N2. So here the investigator has to select delta as according to how many patients the investigator thinks can be recruited in stage 2. Once delta is selected, we have a value for C, and we can choose N1 as the value such that the mean square error on the estimated threshold is less than an upper bound gamma. This is computed by simulation. Once stage 1 is performed with N1 patient, we can re-estimate N2 by simply replacing this formula from the initial guesses. We replace it with the current estimate of model parameter obtained in stage 1 that are then based on real data. In this graph, we observe the behavior of the mean square error of the estimated threshold for increasing N1. The horizontal line is in correspondence of gamma equal to 0.5. For a fixed value of rho B, we can observe that for increasing rho A, so as the biomarker becomes more predictive, the required sample size decrease. So going from the solid line to the dot line. Finally, we have the results for the whole simulation study. So different correlations. These are the values for sample size stage 1 and sample size stage 2. I want to focus your attention on the value for N1, because you may think, and I agree, that they are pretty large for just a preliminary study in which you want to estimate the threshold. But let's investigate more on what happens if this N1 is not large enough. Assume that you are in a clinical context in which you want to stop the trial and not go to stage 2 if the benefiting population is less than 10%. So in practice, you want to stop the trial if this probability is less than 0.1. The idea is that if N1 is too small, you don't have enough information to estimate the threshold, and you can go wrong to the point that you stop the study when you should actually continue. We evaluated this probability in a simulation study. So we consider here N1 on the x-axis, and on the y-axis, we have the simulated average probability of incorrectly stopping the study. That is computed as the sample proportion of time in which the study was stopped in the simulation, when instead it should continue. We observed three groups online in correspondence of rho A minus rho B equal to 0.3 for group A, rho A minus rho B equal to 0.4 for B, and rho A minus rho B equal to 0.5 for C. As the biomarker becomes more predictive, so across the three groups, this probability of incorrect decision strongly decrease. And you can also observe a within group effect. So as the biomarker becomes more prognostic, and so as the magnitude of the rows increase, this probability decreases too. So very briefly, oh, I see something in the chat. Okay, thanks. Very briefly, before I conclude, this is the density of the estimated threshold for increasing N1. And you can see that as it becomes larger, the density tends to be more and more concentrated around the true value, that is the vertical line. So to conclude, we have provided a complete template for sample size considerations for both stages. Since we characterize the correlation using a bivariate normal model, we have shown the direct relationship between the predictive nature of the biomarker and the sample size. The result that we want to stress is that the validity of the second stage largely depends on the success of the first stage in estimating the threshold. So even if the investigator may balk at expending so much resources on a preliminary study, the cost of not doing that is a much higher likelihood of having a less reliable stage 2. So given this insight, one may reasonably question if it's worth enriching. And the answer that we gave is that unless an investment is made in the threshold estimation, the trial will likely lead to the wrong conclusion. And following our approach, if the subpopulation is too small or too large to be cost effective, the study might not be taken to the second stage. Just a few lines of further research. So we would like to extend our model and our approach also to trial for binary and survival outcomes. We would like to include multiple biomarkers instead of a single one for a better identification of the target population. We are actually working on optimal allocation problem for two-stage enrichment design, and inspired by the idea of Zhao and LeBlanc, we would like to introduce a design criterion that also takes into account those patients that benefit from the treatment, but that are not in the subpopulation. And this is a list of references, and I wish to thank you for your attention. I hope I'm on time.
Yeah, we have 1 minute. Does anybody have a quick question?
was very nice.
Oh, thanks, Nancy.
Do we have extra time at the end
Like 5 minutes.
that they'll give us before they knock us off?
Yeah, I don't know how that works.
We'll find out.
We will. All right. Our last speaker is Dr. Hyun.
Okay, thank you, Jessica. So let me share my screen. So I don't know, I tried yesterday, but I'm not able to share my screen. So that was that was why I sent the email. So I'm still not able to share my screen.
You aren't able to?
No, I mean, when I click the share screen, it said the disabled screen share.
Do you want me to do it for you?
Yeah, that'd be nice.
I don't know how to make it full screen mode. Does somebody know how to do that?
There must be.
Control L. Oh,
Like magic. Thank you.
Okay, thank you, Jessica. Okay, so good morning, and thank you so much for inviting me to present my research here today. This is joint work with Frieri, Jamshedian at Gilead Science and Chitra Seung at Campus Labs. So I'm going to talk about how we search optimal timing for an interim analysis for a primary endpoint under some model assumption. Next slide, please. Okay, so in adaptive clinical trial, an interim analysis is often conducted to terminate a trial for early success or futility. In this research, we'll focus on a trial with one interim analysis for early success. Okay, well-planned interim analysis can stop a trial for early success and potentially make a treatment available sooner to patient and providing shorter time to market for manufacturers. So well-planned interim analysis can lead the trial very effectively. Successful implementation of an early success interim analysis very much depends on the timing of the interim look and the amount of the data accumulated. If we do interim analysis too early to save more cost, then we may not have enough information to do the analysis. And if we wait too long to accumulate enough information, then we may not save very much money in the cost. Thus, the timing of an interim analysis is very crucial. Also, I mean, many times in my previous work setting, we try to do interim analysis in the clinical trial, then always the coming back question is why you set the interim time at that time point. But we try to make some justification or reduce the research, but the answer was there was no clear justification for deciding the interim timing. The simplest approach known is to conduct the interim analysis at equal intervals, but there is no clear scientific justification for this, and this cannot guarantee if the study can be terminated effectively. We found some researches for studying the timing of interim analysis, but none of them considered time to event data. And in my previous work setting, we test for safety and effectiveness. Then safety most likely binary event, a binary endpoint, and the effectiveness was most likely time to event data. So that was our the motivation why we want to find out what would be the best timing for the interim analysis for the success for the time to event data. Next slide, please. Next slide, Jessica. Yes, thank you. In this research, we consider a single-arm and two-arm clinical trial for testing a primary endpoint based on the time-to-event data. Then we study the optimal interim timing for an early success interim analysis. Okay, for the time-to-event data, it is in this research, we assume that the exposure time follows a piecewise sequential distribution. The most common parametric model to describe the time-to-event data is exponential model. It describes a survival function over time as exponential decay model. But this model assumes a constant hazard rate over time. But here, piecewise sequential model is more like generalized version of the exponential model, which assume different hazard rate for different time interval. In clinical trial, it is often observed that non-constant hazard rate for different time interval. So piecewise exponential model can provide very flexible model fitting. Okay, one way, so in the clinical trial, we want to maximize power. Okay, and instead of like directly maximizing power, here our approach is, in order to maximize power of the trial, we want to minimize the variance of interest, because if we minimize variance, it can maximize power for sure. Optimal design theory explains how to design a trial effectively to minimize the variance of the interest. So based on this optimal design theory, for the time-dependent data, we examine the variance of the interest as a function of the timing of the interim analysis. Then we obtain the optimal interim timing that can minimize the variance of the parameter of interest so that it can maximize the power of the trial. Here, the proposed method is easy to understand and very straightforward to implement. And this framework, we believe it can be easily extended to other types of endpoint with modeling assumption. Okay, next slide, please. Okay, so for the time-dependent data, there are following information observed for the I subject. So capital Y sub I, that's the enrollment time for the I subject. Capital T sub I, that's the time to event measured from the I subject enrolled. Capital W sub I, that's the time to withdraw or drop out measured from the I subject enrolled. Then here, capital X, that is the timing for an interim analysis measured from the last subject enrolled. And this interim timing, capital X, can take any value between X1 and Xn. And X1, we say that's the earliest possible interim time, and Xn, that's the time for the end of the follow-up, end of the trial. Then capital CI is the censoring time. So it takes minimum value between I withdrawal time and total follow-up time for the I subject. So that's the capital X plus Y sub n minus Yi. Then for the I subject, following data observed. So small ti, that's the exposure time. So it takes minimum value between time to event and censoring time. And V sub I is the event indicator. So it takes value 1 if the time to event is less than censoring time, otherwise assigned 0. Next slide, please. Okay, so now let's talk about how we can identify optimal interim timing for single arm study. So here we consider a single arm trial comparing a primary endpoint P sub E to a pre-specified performance goal PG. Okay, the endpoint of interest here is a survival rate or failure-free event rate at the end of the follow-up. Here, survival function, we assume it follows a piecewise exponential model with 8 distinct time intervals. So for different time interval, we assume different hazard rate. So as you can see in the third bullet, so survival function at time t is exponential function of minus time t times hazard function lambda at time t. Then this hazard function lambda at time t gives a different hazard rate depending on which time interval the time t falls in. For example, if the model assumes 3 distinct time intervals, then we have 3 different hazard functions. So lambda 1 for the first time interval and lambda 2 for the second time interval and lambda 3 for the third time interval. Okay, so here the primary endpoint is compared to a performance goal. Then the testing hypothesis is H null, the primary endpoint less than or equal to performance goal, and alternative hypothesis, the primary endpoint is greater than performance goal. Okay, the good thing is under this, sorry about my dog. So under the model, the parameter of interest can be written as a function of the hazard rate. So the primary endpoint then, as you can see in the last bullet, that's the exponential function of minus summation h from 1 to capital H, the width of a time interval h times corresponding hazard function. Next slide, please. Okay, the power of testing the previous hypothesis can be maximized by minimizing the variance of estimating the primary endpoint. So suppose we have a sample of n IID observation. Then under the piecewise exponential model, following likelihood function is observed. So that's given in the second bullet. Okay, then in this likelihood function, there are two components we need to understand. The one is the capital D sub H. So that is the total number of observed events for interval H, and capital DT sub H, that's the total observed exposure time for interval H. Then from this likelihood function, we can get the MLE of the hazard rate at time interval H. So that's the lambda sub H hat. It is obtained by the total number of observed events over total observed exposure time for the time interval H. Then from this likelihood function, we can get the feature expansion matrix for the model parameter. So this piecewise expansion model has a model parameter, the hazard function, and it depends on how many intervals we have. So if we say a capital H distinct time interval we have for this model, then we have model parameter 8 different hazard functions. Then the feature information matrix for this model parameter can be obtained by taking the negative expected value of the second derivative of the log likelihood function with respect to model parameter. So the information matrix given in the bottom of the slide. Next slide, please. Okay, so variance, okay, then from that information matrix, by taking inverse, we can get the variance of the hazard rate. So the variance of the hazard rate can be obtained by the first flat, the lambda H squared divided the DH. So the lambda H, that's the hazard function for the time interval, and DH is the total number of observed events for the time interval H. Okay, the primary endpoint is a function of the hazard rate. So using a delta method, the variance of the primary endpoint can be obtained by the equation in the second bullet. So that's the summation of H from 1 to capital H, the width of the interval square, times the primary endpoint square, times the variance of the lambda H hat. So if you look at this variance, okay, there are 4 different components: the width of interval, the primary endpoint, and the hazard rate, and the total number of observed events for the time interval H. Okay. At the design stage, most likely we know how many intervals we have, and then the width of the interval. So this one is known, and for the interval, we also know the assumed hazard function. So the width of interval and hazard function is known. Then the primary endpoint also we can predetermine. Then this variance of the primary endpoint, it is totally depends on this one component, total number of observed events, DH. So this variance is a function of total number of observed events for the time interval H. And you can see that the variance then can be decreased by increasing this total number of observed events. Okay. So one thing we can see here is the value of DH, the total number of observed events, is changed by the interim timing, because the total number of events can be increased by having more total follow-ups. In addition, the total number of observed events is completely unknown at the design stage. That's true, right? At the design stage, we don't know. I mean, how many events will experience how many patients will experience the event, and who will experience the event. That's completely unknown and random. So actually at the design, so because of that, because this D sub H is unknown, we cannot get the variance of primary endpoint at the design stage. Alternatively, we can approximate the expected value of D sub H under the assumed piecewise expansion model. So here our approach is, okay, now based on this approximated expected value of D sub H, let's approximate the variance of the primary endpoint. Okay, next slide, please. So here is how we approximate the total number of the events D sub H and the variance. Okay, we assume n subjects are enrolled uniformly in K months. Assume 1 month is 4.33 weeks. Then the enrollment time in weeks for the I subject can be defined as the first equation defined as the equation in the first flat. Okay, recall the exposure time for the I subject is small ti, which is minimum value between the time to event and censoring time. Then here, the time to event and then the withdrawal time for the I subject are again completely unknown at the design stage, right? So only piece of information we know we can expect at the design stage is the total follow-up time up to the interim, up to the interim timing. Okay, so here instead of this small ti, what we are going to use is small xi, the total follow-up time of the interim timing x. So that is capital X plus y sub n minus yi. Okay, so under the piecewise expansion model and the assumed ornament time, we can get the expected value for d sub h at the interim time x using the below equation. So that's the equation in BLED 4, force BLED. Here, the idea is we can compute the chance of time t first in a time interval h because we know the probability distribution for the time t follow the piecewise expansion model. Then we multiply this probability to the number of patients in the risk set for the time interval h. Then we can get the expected value number of events within this time interval. So now the variance at the interim timing x can be approximated by replacing the total number of events with its expected value. So as you can see in the last the equation last slide, now I replace the dh with this its expected value. So in this formula again, you can see that the variance is decreased by increasing this expected value of dh. Okay, next slide, please. Okay, so now I show you how we approximate the variance for the primary endpoint under the single-arm study. So here I extend the single-arm study to two-arm study and see how we can approximate the variance for the primary endpoint. So now we consider two-arm trial comparing a primary endpoint between two arms, the treatment versus control. So for the primary endpoint, again, we use the same endpoint, survival rate at the end of follow-up. And the hypothesis we want to test is to see the survival rate for the treatment arm is higher than the survival rate for the control arm. So here we have hypothesis, and again, the primary endpoint for each arm can be written as a function of the hazard rate, okay, just like we did in the single arm. Then the power of testing this hypothesis can be maximized by minimizing the variance of estimating the survival rate difference between two arms. Since these two arms are independent of each other, the variance of the survival rate difference between two arms is equal to summation of the variance of the survival rate across two arms, as you can see the last slide. Next slide, please. Okay, so similar to the single-arm trial, we can apply the same approach to approximate the variance for each arm. So we use the expected value for the number of observed events for each arm at the interim timing x. As you can see in this equation at the second BLED, we can approximate the variance for each arm at the interim timing x. So the idea is exactly the same as the previous single-arm study. Next slide, please. Okay, so now we know how to approximate the variance for the primary endpoint for the single-arm study and two-arm study. So now we talk about then how we find interim timing that can minimize this variance. So optimal design theory provides various optimality criteria to minimize the variance of the interest. Here we consider C optimality criterion. C optimality criterion is used to search a design when the goal is to minimize a variance of estimated functional moral parameters. As shown in the previous slide for single-arm study, the power of the study can be maximized by minimizing the variance of the primary endpoint. Okay, and we know that the primary endpoint is a functional moral parameter hazard rate. Thus, the C optimality criterion can be applied to minimize this variance for single-arm study. Then based on the C optimality criterion, we search the interim time x that minimize the variance of the primary endpoint. Okay, so one thing we know is the total number of events increases as an interim time x does because we have more follow-up, right? Then variance of the primary endpoint can be minimized as an interim time x approach to the end of follow-up XZ, right? So, I mean, from this defined trial, if the interim timing X goes to very close to the end of the trial, then we have the longest follow-up, so the variance of the primary endpoint can be minimized. Okay. The minimum variance is, so as I mentioned, the minimum variance then can be obtained when the interim timing is equal to the end of the trial. Okay, but this is not our answer. Okay, we want to find out interim timing prior to the end of the study that can minimize variance, right? So instead of finding the timing that minimize the variance, our approach is to obtain optimal interim timing by finding an interim timing X that provides relatively small variance compared to the one estimated at the end of follow-up. So again, at the end of follow-up, we have minimum variance. So our approach is find interim timing X that provide relatively small variance compared to the minimum variance we can observe at the end of a follow-up. Okay, next slide, please. Okay, so in order to measure the variance at the interim timing compared to the minimum variance can be obtained at the end of trial, we use efficiency based on the optimal design theory. So here, first, let's show the efficiency at the interim timing X. So that's the ratio between the minimum variance and the variance of the primary endpoint can be obtained at the interim timing X. So again, the minimum variance can be obtained at the end of follow-up. Okay, so we evaluate this efficiency for all candidate value for the interim timing X in the predefined the range of interim timing X1 and Xn. Then we search the earliest time X, earliest interim time X, such that the efficiency is greater than or equal to the target efficiency R. Here we have to select this target efficiency R, providing a good balance between collecting enough information and saving resource. Okay, since this is ratio, so we compare the variance to the minimum variance. So this efficiency must be between 0 and 1. Okay, and the target efficiency close to target efficiency close to 1 means the variance at the interim time X is very close to the minimum variance. So for two-arm study, same idea is applied. So we know the variance. Okay, the variance of primary endpoint. Then again, for two-arm study, we look for the interim timing X that can minimize this variance compared to the one can be observed at the end of the trial. Okay, so again, using this efficiency, we can measure, we can find the earliest time X satisfying this efficiency for the two-arm study. And if it is greater than the target efficiency R, that will be our earliest timing. That would be our optimal timing for the intent analysis. Okay, next slide, please. Okay, so since we don't have much time, let's just go over this example quickly. So for the illustration purpose here, I provide hypothetical study example for single-arm study. Suppose we have single-arm study to compare 12 months failure-free rate to an historical performance goal. For the study, we assume 185 subjects will be enrolled, and the full enrollment will take approximately 7 months on average. Okay, we assume there is 3 months post-blanking period from the date of procedure. Any events happen during the blanking period will not be counted. So the events happen and follow-up time happen between months 3 to month 12 would be considered for testing the 12 months failure-free rate. This is quite normal for pulmonary vein isolation treatment for AFib patient. The primary endpoint here is the failure-free rate at month 12. So this is during 9 months post-blank period. And our testing hypothesis is given in the first bullet. So we want to see the primary endpoint, the survival failure-free rate at month 12 is greater than 50%. Okay, and we assume the piecewise experience model with the 3 distinct time intervals 0 to 2, 2 to 8, and 8 to 39 weeks. And here we have assumed baseline handle function. Then we can do one interim analysis for early success anytime between week 13 and week 52. So the range for the interim timing could be anywhere between week 13 to 52. And our goal is to find early interim time such that this efficiency of interim timing is between 0.8 and 0.9. Next slide, please. 1 minute left. Okay, so I'll just summarize this example then. So this figure shows the change of efficiency for the interim timing X between week 13 and week 52. So y-axis, you see the efficiency, and then x-axis, that's the interim timing. Then this plot shows that, okay, the efficiency becomes 0.8 when the interim is conducted at week 23, after the last patient enrolled, and the efficiency becomes 0.9 if the interim analysis is conducted at week 30 after the last patient enrolled. So here, based on the target efficiency we set between 0.8 and 9, we can do interim analysis between week 23 and week 30. Then it can provide the variance close to the minimum variance we can observe at week 52. Okay, last slide, please. Next slide, please. So, I mean, this one is just summarizing what I present here. What I want to say is, okay, our proposed method is very easy to implement. So whole our approach is, okay, how we can find interim timing that can minimize variance compared to the one we can observe at the end. So next slide, please. Here, everything is based on this times event, but we can thread this one to Bayesian approach. So the Bayesian approach is using positive distribution, and we see that, I mean, for single-arm trial and two-arm trial, the variance of the primary endpoint under this positive distribution, we can also apply the same idea. Okay. I think the time's up, so this is my presentation. Thank you.
Thank you very much. We have a couple minutes for questions if anybody has any questions.
Looks like a lot of good things are happening.
For sure. I think we lost most of our participants.
So what are you going to tackle first, Rosamarie, in your list of new things to do?
Sorry, Nancy, I don't understand.
Well, you gave a list of things to do next.
Oh, yeah. When Bill was here, we work with also my supervisor on the optimal allocation, like formally proving whether or not the balance allocation are optimal. And but we did use the conditional linear regression model instead of the bivariate normal. I mean, they coincide, but we choose to use the other one because it was simpler for the notation, etc. So we started from that, and but we are still working. So we are assuming now that we have heteroscedastic regression model, and we are trying to derive all the optimal allocation, like the optimal design, a optimal design, etc. And also how to implement it. That's maybe is the most challenging thing.
Yeah.
If you need
because your whole environment is as complex as it is.
Yeah. And
so
yeah.
Rosamarie, I see your topic is very interesting because we do lots of enrichment design in my current position. So but one just suggestion is we do, I mean, many times endpoint is either primary, binary endpoint or time dependent endpoint. So, I mean, do you plan to extend your research to like right now it's continuous variable. So like
I think those were
variable or
yeah.
So
Yeah.
that would be really nice.
Yeah, no, it's on the list, and I think one of the co-authors, Jean Tao, is still working on something on the binary model, but yeah, we need time to work also on that. So the list is long and the topic is very interesting, and I'm glad you're using that in your company. So
Yeah, we did a lot. We do a lot.
Oh, great. Okay, so it's not
Yeah.
something that we
That's
just.
good to know because there was some questions about how much it's actually being used.
But
Interesting.
what I see is whenever we do an enrichment design, that's for the feasibility study. So we use very small sample size, like 80 or 90. They don't want to spend more than 100 patients for that.
Oh yes, yes, I know. And it's hard, and like
Yeah.
yeah, when we see that the stage 1 sample size were like 200 or more, we were shocked. But unfortunately, if then you want to see the property, or I don't know, of the threshold, the mean-square error of the threshold, you realize that you will get very large values.
Right.
So sometimes it's also related to the maybe Nancy's maximum tolerated dose. For example, you have usually set the maximum tolerated dose with a small number of patients, and then if you obtain wrong results, it may jeopardize all the entire study. So yes, it's hard, and I'm sure in the applied field it's much harder to spend money on that.
Yes. Always we come with some like moderate sample size, but they say no, we don't want to spend more than 100.
But it's the same problem. I mean, the problem keeps coming that, everybody wants to put their money at the end. And if you get to the end with the wrong formulation, you've just wasted all that time. So at least Rosamarie's work provides a tool to show what they're risking by
Yeah.
doing small sample sizes early on.
Yeah, so it was interesting.
Yeah, I mean, you can take your practical example and evaluate
Oh, yes.
what
Yeah.
the trade-offs are.
Yeah, it's a trade-off. Yeah.
And Jessica, thank you. I don't know. I think this is because of my company computer. They don't really want to share this like environment with any others. So many times I try to use Zoom, I'm not able to like share my screen. So I don't know why.
It's probably, I hadn't thought of that. It probably is your institution that's got you blocked.
Uh-huh.
Well, I appreciate all of you for joining, and we appreciate the Caucus for Women in Statistics putting this session together.
So what happens to the video?
Mm.
It belongs to WNAR, or we get copies, or?
Hold on a second. Let me